welcome to Living Science. Today we have with us Dr. DrKhanna of ICGB New Delhi. Dr. Khanna is among the very few scientists in India who have been able to take their research out of their laboratories to the society. Several technologies that are developed in his lab are now part of the market in the form of diagnostic kits. One of his most celebrated product, which is also locally relevant in Delhi, is the dengue diagnosis kit that has already been in market for several years. Dr. Khanna has received a number of awards in recognition of his work. Ranbaxy Research Award in Pharmaceutical Sciences by Ranbaxy Science Foundation, Biotech Commercialization Award by the Department of Biotechnology, Government of India, Vasvik Industrial Research Award in Biological Sciences and Technology, just to name the few. Dr. Khanna is also the fellow of Indian Academy of Sciences and National Academy of Sciences. Currently, his research group is involved in developing a vaccine against dengue and looking for a compound with antiviral properties from natural resources. Today we are going to have a small presentation by Dr. Khanna on his research work, and it would be followed by a brief interview session too.
Yeah, I think it's now slowly becoming more fashionable to translate your research findings. So, a lot of funding is available, and not only the funding, the hand holding, the expertise, and exposure. I would say DBT, as I said in the beginning, is like has provided all the oxygen to this biotechnology, and it is up to us to differentiate between biology and biotechnology. And so there is a lot of funding available for doing biology. There is always a funding available for biotechnology. And whatever you are doing, if you always have a lens in your hand of so what next, I think it will keep you on a track. And that's what we do in our lab, that we always begin with the end in the mind, and then do a lot of which we call quick and dirty experiment to find out, to test your hypothesis, that are you doing the right things, and if it looks like, and then it's a lot of collaborations, because you cannot, in basic science you could still work in isolation, but in this kind of work you need to kind of interact with several experts from, and you have to try new things. And DBT now encourages multidisciplinary funding, even bilateral funding, between two different nations have been, I would say, a game changer. For example, for diagnostics, it was in the same room where Professor Bhan had brought, I would say, 10 scientists from Finland, and we 10 people from within India, and everybody made 10-minute presentation. By evening, 4 of them were writing projects together, and I have been fortunate since 2006. I have a continuous funding through this Indo-Fin collaborations, and there is an Indo-Fin Diagnostic Research Center which now is at THSTI. And I think what I learned, as I said, that without DBT I would have probably never come across those wonderful scientists who are so focused on diagnostics. Same way with process development, DBT got us in touch with Germans, and they are the best when it comes to fermentations, and all the clones which I mentioned about hepatitis B, making 7 grams of protein per liter of culture. That's unbelievable. Insulin secreted out 3.5 grams per liter. Again, thanks to DBT for making those connections with us. So I think there is if you want, there is a lot of support available, expertise available, and I would also want to emphasize the support available to us through Indo-US Vaccine Action Program. And all these things are driven by Department of Biotechnology. I think this is a global trend now. So lot of people are thinking that okay, there is a lot, for example, repurposing of drugs. The drugs are known now, there is a lot of interest that could we repurpose them, because developing new drugs have become like so expensive. So people are finding out ways, funding is shrinking, they're finding it out ways that to become more useful, to have a more public health impact, because you are spending taxpayers' money. So I think it makes sense to give back something. Yeah, no, this is a loaded question and I don't know how to answer this. I can only briefly say that I had been very fortunate. When students, many of them had come to our lab, we actually discourage them from joining our lab. We tell them that, this is not for you. You could go ahead and do a kinase phosphatase somewhere else. And even then, if they come to you, they are self-driven. So I have had students who have not published their work for 7 years, but they have patents. So I think this is a very, this person, Rahul, who stood first in all the CSIR exam, that Shama Prasad Mukherjee, he was number 1. He came to our lab with this determination that he has to do something useful. And we kept telling him that you please go to another lab, but he still keeps coming to our lab. He will do all the rotations in our lab. And all the students which I showed you, not even one of them was an exception to this rule. And many of them had admission to many places because they are bright kids. But along with that, I think they had some determination. And PhD, I think, is the safest place where you can take risks. Because I give you an example. Hepatitis B, my first PhD student from Bulgaria, she was here. There was a Nature paper which said that in Pichia pastoris in yeast, you can make 300 milligram protein per liter of culture. And it said a line that increasing the copy number, gene dosage does not make any difference. But there was no data. So we discussed, I said, well, it should make sense that increasing the gene dosage, which means increasing the message, you could increase the protein. And whenever you make a recombinant proteins of medical use, the cost comes from downstream processing. If you have to do 100 liter fermentation purification versus 10 liter, it makes a big difference in terms of liquid handling. So we gave her this project that could you make 1 copy, 2 copy, 4 copy, 6 copy, 8 copy, 10 copy, 12 copies, 16 copies, integrate into the yeast and look at what happens. She brings that paper and she says, well, this has been reported in Nature that it does not make a difference. Then you say, where is the data? It's just a line. But then they feel that we don't want to work on something negative. So then it's not negative. What will you say? That increasing copy number does not make a difference. That will be one most negative thing. But it is publishable because there was no data. Or you could also imagine that increasing copy number increases the amount of protein up to a certain level, then it decreases. That's also a new information. So what you need is information which you can publish. And when she understood this, that I am in safe hand, it does not matter, I just need to produce this data and it will be published. She used to work almost 18 hours a day and self-driven, finished all her work in 1.5 years. She would be in the lab even 2 o'clock in the night, and if you have entered ICGB gate, if it, it's pretty height. So she was telling me once that it was raining heavily and she has to take a sample at 2 o'clock in the night. She put the scooter outside, climb the whole thing, go to the other side, do the experiment, go back, and it was raining and she hit a cow and she broke her hand. But next day morning she was here with again. So some of these kids are self-driven, and when we published that paper, and we were surprised to see that there was an increase in the message, there was an increase in the protein, and this was my first student, and that clone is the one which we were able to sell it to Chinese.
Insulin.
Hepatitis B, yeah. and then Professor Barale from ICGB Italy, they wanted to work on insulin. They had a weak clone for expressing insulin. So we followed the same assembly line, increased the copy number there. We had more things there. So I think PhDs are very safe place to take risks. You just need to kind of be very critical. You just need to be very analytical, and don't worry about the outcome, what the outcome would be, because whatever is the outcome, you could publish it. I agree and disagree with this statement. You see, you cannot compare IT with BT. In IT, replicating same thing is so easy. In biotechnology, taking something from clone and taking it up to the clinical trials and putting them out are two different things. We just cannot compare them together. And I think within India and outside, biotechnology had made a huge difference. And you would see many of these blockbuster antibodies or recombinant proteins of medical use are all fruits of biotechnology. And I showed you many diagnostic kits and vaccines. Means the impact of a vaccine, you can imagine. So I think if you think that would it become like Infosys and this and that, I don't know, but I don't think you can compare both of them. But whether it is you go to Finland, I went to Finland, and they, as I said, the PhD students do their postdocs in companies. They have a startup facility within the university. I wish I could show you the pictures of that. There were rooms which you could rent, which had a screen here with a remote, and there was like bean bags. You could lie down. There were no tables and chairs. You could have different kinds of room that some had just beds in them. You could just, it was like a different way of kind of leaving them free, to think. And students could get into any one of these rooms, rent any of these places, open their companies, explore their ideas in real sense. Many times when we want to open something, let's say in a biotech park, we say, what is new in this? And sometimes the minute they start to work a little bit, we say, okay, move out from here. I went to University of Wisconsin. Their biotech park was 3 times the size of Jawaharlal Nehru University. 3,000 acres of land completely filled with companies who would make reagents which are used in biotechnology, which we order. And they have been there for 17 years, and nobody is asking them to move out. Then they made phase 2, phase 3, So I think with what we have limited resources, within that we need to generate our own success stories and not always become a drain on the government that they should provide everything. And I think we have the cheapest education possible in this country. You go to any other place, if you're doing engineering or medicine, you are probably done by, I don't know, $500,000 as a loan. So government is doing more than what they can do. I completely agree with you. Things have changed. In fact, when we were doing our masters in biochemistry, and I still love that, it was 8 hours of practical every day, half an hour of theory. Now it's the other way around. It's probably half an hour practical and 8 hours of theory. If you do your things with your own fingers, you know what has happened. And I don't know why is that change. Maybe there is less funding. In many places you are shown demonstrations, rather than you doing the experiments. A tissue culture is shown just like a flask. I'm sitting here, I'm just showing you a flask and a pipette inside. You have not done any of those things. And so maybe the number of students have increased, but the funding probably haven't increased in that ratios. I don't know what the answer is to that, but I see your point that where I did my masters, when I go back to that place, it's the other way around. Now there is a lot of theory and there is very little practical. You go to University of California, they will teach you something, but they will ask you something different. If we did the same thing here, all of you will say you out of syllabus aa gaya and hartal karoge bahar, because we want to be spoon fed. We should ask ourselves also, ye to out of course ho gaya. Do you agree with that or no? I don't know. Yeah, again it's resources. In many places high school kids come to universities and labs for their summer training. We even refuse undergrads within, let's say, many institutes. We say, oh, there will be too many people, we don't have infrastructure to absorb them. We select probably 10 out of 1,000 of them. So it's a policy of rejection rather than acceptance. And I would imagine that more resources for education should be available. No, I would even engage in high school boy, because they think very differently. They think very differently. This multi-epitope protein means, I didn't have the time to share. Everybody would make a small peptide. You are a chemist, you make those peptides. And this person who came to us from Goa, he said, well, why can't we just link them together, and ask bacteria to make this protein? We never think of those because nobody has done them before. And then you put your thing, oh, you are stitching epitopes and things are changing, and then of course things would change. Then you figure out that could you put just plain glycines or something. So if I show you one protein here, I have probably made 19 proteins, not I means students have done and looked at the combination. So I think they bring something which you have never thought of it. And it is very important to have those kinds of students come to your lab who have never thought, because they are not conditioned on those. Yeah, I would say it was very easy, because ICGB is a very unique organization. I'm a group leader of a called Recombinant Gene Products Group. You go to any institute in this country, there is no group which is listed as Recombinant Gene Products Group. Right? So my promotion, my evaluation was only done that how much I collaborate with companies. So it wasn't very difficult for me. And one of the mandate of ICGB was the biotechnology for developing nations. This was the mandate of ICGB, and if that's the mainframe mandate of ICGB, we were never questioned, why are you doing this work? So, and I think, as I said, that the circumstances were right, that companies were coming to us, but then what we gave it to them was something so good that they kept coming back to us. And if they came to us saying that this didn't work, we worked with them to kind of make sure that it worked. And sometimes you succeed, sometimes you fail. And you only talk of, if your 10% of your experiments are working, you are doing great. It looks like that every experiment you do works. That's not going to happen. So every protein which, if I show you just one protein, that's not the protein. There are several other versions of this which have gone into it. And this is where the students, can multiplex, they do this, and then you give these free samples to these companies and say, okay, is this right or is this right? It's like, when you go to check your, glasses, that is this better or is this better? You don't know which one is better. They tell you which one is better, and then you keep moving in that direction. So I think that communication with the companies and faith with the companies, and also having this thing that they are right. They know what works. You cannot say, many times what I find is, oh, I have transferred it, now it is their problem. No, it is our problem. That's where is the difference. Any experiment which you either get outsourced or you give it to somebody else, please do that experiment in your own lab first. First experiment has to be done with your own student. Look at that result. So you know what you are doing. Don't believe in that you gave it to somebody and say it doesn't work. If it doesn't work in your hand, it does not work. But if it works in your hand, it works. And if it doesn't work in their hand, you have to figure out, you have to work together side by side, and things come up. So So I would say first thing is that do not procrastinate. If you have something, do it now. We say, oh well, this thing happens, that happens, maybe unless I have this. For example, a lot of people think, oh, if I had this antibody, then I will do this. Tell them, you have this antibody now, tell me what are you going to do with it. Many times you will feel, I don't need that antibody, I can do without that antibody. But we know after spending 2 years getting that antibody that there was another way out. So I think question yourself that do I really need it? Is there another way out? Talk to people and talk to new people who are not in your field. They will bring such new ideas. This multi-epitope protein, I told you, people would have been making peptides, and we were also doing at ICGB, a lot of chemicals, fumes all over the place, and here bacteria does it free of cost for you. And also believe in your results. Sometimes we don't believe in our results. We say, ye kya ho gaya, like this is not me. So that is very important. We had a student in our lab we sent to Germany. We had a clone which was making, let's say, insulin 300 milligram per liter. We sent this student there, and his first experiment, he made 1 gram per liter. So, we were very happy. 1 gram, it's like 3 times. Then 15 times it was 300 milligram. Now all of us will say this 1 gram was wrong, right? Because you could make a mistake, maybe measure it wrong, but not that German professor. She said, but you got 1 gram. And we kept saying that, but you got 1 gram once. 15 times you got 300 milligrams. She said, no. And it is the 16th time now they were getting 3.5 gram. Because, and then they figure out that there was glycerol or methanol, whatever they were using, there was a copper contamination which was killing the cells and it was not letting them induce. Once that stock was over, it started to work. So having that trust, you see that difference? Means I would tell my student, you made a mistake, just trust with your 300 milligram. But so I think looking at your data and having that, if you got something right, and this is where the documentation is very important, that how much did you do, what did you do, and when they do the fermentation, it's like neurosurgery. Around the fermenter, there are 9 people who are taking every 20-minute samples for carbon, for nitrogen, for phosphate, for trace metals. What we do is, because lack of resources, we put our E. coli in the fermenter and just walk away, have a cup of tea, come back and think that everything would happen. That doesn't happen. So again, this collaboration established through Department of Biotechnology, really, go to people who have done that job before. Get a jump start. Sometimes we want to do everything ourselves, and that delays, and time is money. I think every Friday we have our lab meetings, and there everybody understands that you have a race against time. If you are making a dengue vaccine, we are 20 years or 30 years behind than anybody else. So if somebody is doing a good job in scoring virus neutralization, everybody does not have to do this virus neutralization. You bring the sera, give it to this person, let him do the virus neutralization, because it's like an assembly line. It happens, you get the results. You look at this, change your protein, do whatever. So I don't know, maybe these students, they do understand that the competition is not within, it's outside. So I guess if somebody has to do immunizations, for example, sometimes our animals are not of that quality. So we have to get the immunization done in, let's say, in Bangalore, in some company. So there is only one person who goes from our lab, Upasana. She would carry everybody's immunogen. It's not that it's mine and yours, because the same person, she knows what she's doing, intramuscular injections of 50 microliter. She is an expert in that. If you have to do some score pathogenic antibody, you have to inject immune complexes into eye of a mouse, which is very difficult. But there is one person in the lab who does it very well, so everybody gives it to him. So I think we have established those things, that nobody owns everything. Together we own these things, and our competition is with outside and not within us. Yes. So again, I think Dr. Bhan has been a source of inspiration to me. He was also from AIMS, so maybe he had a soft corner. And he realized that we have some strength of developing diagnostic kits. The celiac test, which tests for wheat allergy, it comes from Finland, 22 pounds per test, and it's very difficult to do a test at that much money, but AIMS was using it once in a while, unless. So they asked us that could we kind of make a test for celiac. They had the sera, patient sera available with them. We had the cloning, expression, purification, trying to make those recombinant protein or the antibody available with us. We go back to the company, J. Mitra, they had all the optimization processes available with us, and THSTI. So this was a multi-institutional collaboration where our part was to just provide the heart and soul of this. So we joke in the lab, we don't like happy people, fine. So I guess now a lot of my work has moved to other places. For example, THSTI, one of my previous student is a faculty there, Gaurav. So a lot of diagnostic work, for example, this multiplex diagnostics are being done with them. So I have a less load of that. He takes more load of that. I'm just focusing on dengue vaccine because that I think needs all the attention. So I believe that if you work smart, you can save a lot of time. I give you an example. I was in San Diego in a lab where in one lab there were 35 postdocs, right? It was a factory, and if you go in that lab, you feel like zero, you feel scared in that lab. Everybody comes, and my boss would be 22 hours in the lab. I would go to the lab at 6 in the morning, he's already there. So I want to be before him. I go at 5 o'clock in the morning, he's already there. So I can't go before 5 o'clock. And he will go back home maybe 3 o'clock, come back at 5 o'clock. And he was a crazy man. He was like the youngest professor at University of California, and his hands would, he could not put a pipette and a tube together. He was under so much stress. He will publish 35 papers a year in high impact, but he would be so unhappy with him. And he would tell me, I was purifying some transcription factor, and whenever you show him anything, he was always very rude. What is this shit around? What is this around? And I thought, and I was also working like without thinking, 14, 15, 16 hours, and you have a family to take care. One time we had a major fight, me and my boss, and then I used to follow his instructions, because once you go to a new lab, whatever they tell you, do it. So once he started to question, he said, who asked you to do this? I said, you asked me to do it. He said, you don't have to listen to me. I said, this is the last time I listen to you. And then no good morning, no good evenings. I knew I'm paid for 8 hours. I'll go to the lab at 8 in the morning, leave by 4 o'clock. He was so unhappy now, he didn't know what to do. But those 8 hours, I would not take a lunch break, I would not go for tea. I knew exactly what experiments have to be done. I was not seeking anybody's approval again. In 3 months, I purified that transcription factor, which they were not able to do for almost 6 years. In this lab, PhD student will take 8 years, and so it was but thanks to him, once I leave my lab, he would look at my lab notebooks, and as I said that we write very well. He doesn't need me. And I salute him that after 4 months, he came home to my place with his wife, with his kid, with a bouquet of flowers to apologize, to say, Naveen, people who work 16 hours, 18 hours, you produce twice that much work in 8 hours. And he said, I was wrong. And we changed those things. So I think you can really create a balance if you really are dangerously organized. You know what has to be done. Sometimes we keep telling our students, you don't need hard work, you need smart work. Find out, many experiments, as I said, oh, I need that antibody. Tell them, you have this antibody. Then they say, no, I don't need this antibody. So you save lot of time on many of these.
So what I wanted to ask you is that you have a very you have a sorted workflow. You do the lab work, you prepare the vaccine candidate, but you did not go all out into the world saying, now I'll open an industry which will constantly manufacture vaccines and stuff like that. Was it because of the times that you started in which did not support this microenvironment? Now with a lot of support from like BIRAC and all that, where they allow you to be an entrepreneur yourself with your product, if you were to start today, restart today, would you go the same way?
Yes, I think I would go the same way. The reason being that you can't be doing everything what these diagnostic companies have done and fought wars. I don't think I have the, I realize my limitations too. You must have a lens in your hand. If you have so much sunlight, nothing happens. You take a lens, you make a difference. So I think you are just one small part of it. As is kept repeating that a lot of success is from other people, but we don't keep asking what is in for me. I think ICGB gives us good infrastructure, good salary structure, and it's good to see your things outside in the market. One of my student, her mother suffered from dengue in Karnal, and when she goes to a lab and when they do the test, it is our test, and she's so happy to see that. And so she will call me from there with the picture, this is, and of course mother is dengue positive, but and market is very harsh. Last year I think the government of India tried, there was a lot of rumors they will ban this test. They think this test is bringing panic in people. They think that this test is false positive, because as I said, other tests are not picking it up and this one is picking up. So they think that this is false positive. Then you would see that I was told that there was some Chinese dengue test which appeared in the market. You don't need a serum, you can put urine on them and they start to show positivity, false positivity. So there are a lot of issues which happens which only business people who are doing those things can understand. You can only be a small part of it. So I think I would exactly do the way I have done it.
A related question to that. Sorry for it being a prolonged one. Being an Indian, the drive is always to give back to the country and give a test which is as economical as possible, because we have faced it, right?
Yes.
How do you ensure price point control, because you're already giving license in the
We
patent?
don't ensure price because I think companies are smart and we should never interfere in their business. We don't know how to do business. How should we tell them how to do business? I gave you an example of dengue. They launched at 350, they have brought to 160, and trust me, he tells me that it's a sliding scale. He can bring it to 120, right? But if you sell it, let's say for 50 rupees, they say maybe it doesn't work. So there are different forces, and I think they understand it better.
Very nice. I have a couple of questions. We in educational institutes, we look at the model of MIT and Stanford. We start to think that the IPs that's generated in educational institutes in some way contribute to the monetary backbone of the institution. And we in IIT, we are starting, and so we are debating on how do we make a policy out of these things. So there are some things like exclusive licensing and or a single turn licensing or a revenue based licensing. So can you comment like some few suggestions from your own experience that how do you ensure we as an inventor, we are interested in making sure that the innovation succeed?
Yeah, so many of our diagnostic tests have never been given exclusive. If you're giving something for $150,000, you can give it to 2 people or 3 people or 4 people and multiply $150 into 2 times, 3 times, 4 times. Diagnostic industry is a weak industry. They do not have, unlike a pharma or vaccine industry, they don't have so much money that they can give you huge upfront and everything. On the other hand, the vaccine which we have a several $1,000,000 agreement. Now, if it is several $1,000,000 agreement, that has to be exclusive, because the company is also going to spend several $1,000,000 on its development. There you cannot say, oh, I give it to somebody else also. So, I think it depends on the value of the technology, its market value. You know that vaccines can sell in billions, you know. So, many of these decisions are taken within the institute, and we hire patent lawyers who understand these things, because many of us, we do not understand. If you give me an agreement which is signed between Sun Pharma and ICGB, it will make my brain heat up, because there will be so many words in that I would not understand. So, there we take external help. I went to Wisconsin University, and they have some system that any patent which you have or information, you have to disclose it to this university lawyers, and there is a set of people who would decide that do we want to pursue this, and if the answer is yes, they will take away your process, they will file the patent, find the collaborator, do everything, and of course, you will get the return. But if they decide that we do not want to file this patent, then you are free to spend money from your pocket to patent it and do whatever you want to do with it. Many times what happens is that patent must generate money, because if you just have patent for the sake of patent for your CV, it is not a publication. Some of the vaccine patents could cost you like $400,000 to maintain. It must generate several times of that money for this justification. You cannot be like a drain onto the system. So, I think these decisions have to be taken by the PIs and by the companies. This is where the company involvement is very important. They will only work with you for major thing if you have an exclusive patent. If, for example, my student would have published this work of dengue vaccine and I do not have a patent, Sun Pharma would not come with a several $1,000,000. But on diagnostics, we do not care. We publish, but as I said that what we publish, you will see in our paper the sensitivity is 97%, which is good enough for publication. Specificity is 100%. But for product, 97% is garbage. It has to be 100% sensitivity or 99.9% sensitivity. So, we have a different version of the same protein which has a higher sensitivity. So, we do publish our earlier versions for PhD thesis, but the actual one which is being used in that remains with us, that remains with that company. Whosoever gives us the money, he gets that information. So, this is how we kind of balance that.
So, one more on the same topic. You had multi-institution collaborators, and in some of these things, when you are filing for patents and other things, you have any interesting experience or suggestions to add on?
Yes. So, we had a welcome trust project, and they, I think, had your soul. So, they kept telling us, be very aware when you work with several institutes. For example, our non-human primate experiment were done at Emory. Now, if we have sent a vaccine, they have just immunized monkeys and given us the sera, that does not call for a name in the patent, right. If we have outsourced something to somebody, that does not call for a name in the patent. So, welcome trust guided us that it would be that student who had done that work, and it would be me only for the vaccine. For rest of the others, we have several like publication, I have 6 students, 10 students, we were not aware of it. Most of them were within ICGB or a member country of ICGB, for example, somebody from Sri Lanka and things like that. But for vaccine, they kept a very close eye on this just to avoid these kind of things, that who owns the patent. So, it is ICGB who owns the patent, and 2 investigators from ICGB are the inventors of that. So, this knowledge came to us much later, and I think this is very important, and Sun kind of like that. If we had Emory also part of it, if we had somebody else, a global vaccine part of it, they will wash their hands away. So, you must, that is why I am saying you must do those experiments yourself first. Now, we had immunized mice, we knew the neutralization titers. Mice and monkeys are okay, they are not that different in terms of immune system. So, if somebody tells me, oh, it did not work in monkeys, I know it will work in monkeys. So, you can go, but suppose we did not even do the experiment in mice and somebody, it does not work in monkeys, say, okay, let us close down the product. So, and again, you know, when Sanofi is working on vaccine, Merck is working, GSK is working, NIH is working, Takeda is working, several players on this, it is very difficult to convince funding agencies that you have another vaccine which people have been trying to make for 20 years. And when we applied this project to Wellcome Trust, there were 307 projects globally competing for this, 6 were funded and this was one of them. So, you know, that also brought a lot of confidence in us that we are on the right track. So, we think differently. If everybody is doing this thing, we do not do that. Everybody is making viral vaccine with the whole virus, we take away, we think less is more for dengue vaccine. We just take the non-immunodominant epitope, make it immunodominant through vaccine. So, I think once again interacting with people, but keeping that in your view that the patent should remain with the institute. You can share publication. Now, we are writing a paper of that, there will be 20 authors in that. Your University of North Carolina, Emory Vaccine Center and global vaccines and people from Singapore, everybody would be an author, which is okay.
Actually, that's a very valuable, I have made that mistake, so I understand the cost.
Yeah.
That you have to be very closed when it comes to patent.
Yes.
Really, it's a really realistic suggestion you have given us. If there are more questions, if not, then we can call it a, and we can thank Naveen for so much of his time on a holiday.
Yeah, all of you, and I think